Well, hello everyone and welcome to today's webinar. My name is Dawn Van Dam and I am the president of 1 Million Solutions in Health. So welcome to our webinar today. It is called Incorporating Safety Pharmacology Endpoints in Toxicology Studies. And what we're looking at is transposing the experience with biopharmaceuticals into small molecules. So I'm really excited to be presenting this today, but I'll give you some background information on our talk and our speaker, and then we'll hand it over to our speaker for today. So the background to this presentation is, while the science and practice of preclinical safety assessment remains remarkably similar to what was performed 30 years ago, there are significant evolutionary changes that have been implemented in the past 5 years. Currently, these practices are summarized in 2 International Conference on Harmonization documents entitled ICH S7A and ICH M3R2, which present safety pharmacology and general safety assessment respectively. The General Toxicity Guideline was issued in 2010 and contains several new elements, and the Safety Pharmacology Guideline is being actively reconsidered. In this webinar, the need and justification for standalone studies will be discussed based on the progress and experience gained in the last decade. Dr. Beatriz Silva Lima will also discuss the drivers and considerations around the changing of the ICH S7 guideline based on current thinking and results in this field. This should be an exciting and educational session that should have thought-provoking ramifications relative to modern preclinical safety assessment practices. So we're very much looking forward to this presentation today. So let me just tell you a little bit about our organization, 1 Million Solutions in Health. As you may be aware, our goal is to improve healthcare, life sciences, and the overall well-being of patients and consumers, but doing that one solution at a time. So we offer many different ways to do that, and we're excited to be bringing this service to you. So we are a not-for-profit organization, and our goal is to help people connect. So perhaps you're sitting there in New Zealand, and someone is sitting there in France, and someone else is sitting there in Brazil. And really what we'd like to help do is put you together to improve healthcare around the world, to accelerate life sciences research, and share patient and consumer-focused ideas and solutions. We're really trying to build a terrific community. Today we have over a couple million members that are involved in our community, and we're really trying to evaluate new science and technology initiatives and bring them forward. So we would encourage you, if you're in academia, government, if you're an entrepreneur, if you have a business, if you're a consumer, and to share with us your ideas, excuse me, that we can also then share with the global community. We're very excited to do that. We also uniquely have a technology evaluation consortium where we bring new science right to the pharmaceutical and biotech industry, and through them, they help see what are the best tools and technologies and solutions to help them discover new medications and life-saving cures for the diseases that are involved today. So if you would like more information about our consortium, either to share your science or share your technology or your service, or if you're a pharmaceutical or biotech company, or someone from government like the FDA, the NIH, or another organization of that sort anywhere around the world, we would love you to join that consortium as well. So there'll be a survey at the end, very, very quick, only a few short questions. We would love you to fill out that survey. Let us know your interest in future webinars. Let us know your ideas, and also let us know if you'd like information about how to participate in 1 Million Solutions in Health. So let me give you a bit of background about our speaker today. Dr. Beatriz Silva Lima. She is a PharmD and PhD in pharmacology and is a full professor of pharmacology and pharmacotoxicology at the Lisbon University Faculty of Pharmacy and head of the Department of Pharmacological Sciences. She is the coordinator of the research group Pharmacological Sciences of the IMED-UL, Research Institute for Medicines and Pharmaceutical Sciences, where she also heads the research group in non-clinical safety and regulatory science. Dr. Beatriz Silva Lima has 20 years of experience as an expert in non-clinical and regulatory science at InPharmed, National Authority of Medicines and Health Products, and EMEA. She has been a member of the Committee of Human Medicines, Committee of Advanced Therapies and Scientific Advice Working Party. She has been the chair of the Safety Working Party and has been involved as co-deputy in ICH discussions on guidelines on behalf of the European Commission. She is also an independent consultant on non-clinical drug development strategies, and she also acts as a non-clinical expert and member of the NDA Advisory Board. Since this January, she also chairs the Scientific Committee of the Innovative Medicines Initiative, or IMI. And her areas of research, and she is interested in, are regulatory science, non-clinical safety, pharmacology of metabolic diseases, smooth muscle contractility, and associated cascades. So, with no further ado, I'm very excited to turn the baton over to our speaker today, Dr. Beatriz Silva Lima, and let her take you through this exciting information about considering where we're going in the future. Over to you, Dr. Lima.
Thank you so much. Good afternoon, everybody. So I see we have 30 attendees on board, and it's quite exciting to be here, sitting in Lisbon, or more appropriately in studio, close to the sea, and thinking that we are really spread and thinking about, let's say, the future in terms of regulatory changes for drug development, I would say. So yeah, the issue, the objective of this talk, I would say somehow, is to bring a bit of discussion, and again, the discussion which is on the table now with regards to the future and where we are in terms of drug safety assessment, and in particular now the aspects related to safety pharmacology. I would say that the title of this presentation, by referring, transposing the experience with biopharmaceuticals and small molecules, somehow I would say currently it goes beyond that. It's more than transposing what we know or what we got from biopharmaceuticals, but it's more what we are getting from the environment where we are living now in terms of technical and scientific challenges that are growing immensely currently. So just to focus on safety pharmacology and where the discussion comes on whether safety pharmacology studies, the way they are being conducted now, they are really, well, so self-sustained that there is nothing to do to change them, or whether there are possibilities to incorporate or to think on new models that could allow a different insertion on the paradigm for safety assessment of new molecules and drugs, which is now being challenged all the time with regard to its format and to its efficiency. Safety pharmacology is not such an old concept. It started more or less, emerged for the first time in 1980, and up to the 90s, where the concept first was expressed on a guideline, and it was the harmonized guideline ICH-M3. So it was requested that studies addressing the potential effect of pharmacology in organs in general or in human systems, it was needed. So in vital systems it was needed, but the definition itself and the request of safety pharmacology studies emerged in this guideline ICH-M3, which was published in 1997, referring to timing of non-clinical safety studies for conduct of human clinical trials for pharmaceuticals. And at the same time, more or less, the guideline on non-clinical safety studies to support the development of biotechnology-derived pharmaceuticals, which were at that time very much, let's say, empowering and growing, so have been also considered. And in both guidelines, what was referred is that the studies should be conducted to support the use of therapeutics in humans, and detailed studies include their definitions. Well, they were left for future discussion. Somehow the concept was there, and in ICH-M3, it was like a definition by saying safety pharmacology would include the assessment of effects on vital functions, description that they should be cardiovascular, central nervous, respiratory, and should be evaluated prior to human exposure. So somehow it was this consideration that, yes, you have a drug, you don't know what will happen when you go to give it for the first time in humans, it's a new thing, so let's make a kind of a approach looking at the potential effects on the vital systems in general related to the mode of action, and it was related therefore to the pharmacology. And but anyway, at that time, even from the first start, ICHM3 is saying already this study, these evaluations may be conducted as additions to toxicity, or they may be conducted as separate studies. So the concept that you don't need to dissociate completely safety pharmacology, some other toxicology studies, has been there from the start, I would say, of the request when it started to be standardized in guidelines. Then in 2001, a bit later, these discussions that were referred in ICHM3 that, well, they will be left for later, yes, they have been consolidated also in the guidelines, yes, 7A, that we all know very well, and which then, yes, bring the definition as the studies that will investigate the potential in undesirable pharmacodynamic effects of a substance on vital physiological function in relation to exposure in therapeutic range and above. Meaning that, well, yes, it should be the pharmacology and should be the pharmacology-driven adverse effects, which somehow, the more I look at, let's say, this information, the more I think, the more I see that even these concerns have been evolving a long time. Because if you think more and more, what you have in terms of safety concerns for most of the new drugs are more related to pharmacology-driven effects than pure toxicity in the concept related to cell cytotoxicity or whatever. So those really that are the big challenges now for the prediction are really those which are associated to the pharmacology. And I would say this is more and more true. I would like to know whether the colleagues are in agreement or not. So, and indeed, these systems which are listed in the guideline, the cardiovascular, the nervous, the respiratory, are really the first and the most relevant. And if we think well, it's also with the exception of hepatotoxicity or other types of idiosyncratic toxicities, really they are the systems which bring more concern. These studies, as requested by ICHM3 or requested by S7A, they should be mandatory before the first in-human administration. And they should be conducted according also to the requirements on S7 and on any toxicology guidelines that they should be conducted in relevant species. And again, here we have a kind of, well, not very easy way to deal with these studies because they have to be conducted before first in human. And we know that first in human studies are to confirm pharmacology or to confirm pharmacokinetics, but they have to be conducted in relevant species, which, well, in principle, presume that you must know somehow the comparative aspects related to humans, which I would say currently is more possible than it was 10 or 12 or 20 years ago. So, factors for relevance, now we know them very well. The pharmacology, that should be similar in the human being and the animals selected, and the pharmacokinetics, and this would impact on the strains and the species and the gender and whatever. And I would say somehow there is a lot here that would not be, very easy to get in anticipation when conducting the studies if we think that they are needed before the first administration in humans. And then, indeed, the guideline, the S7A, refers to that and recognizes, well, we need relevant species. But during early development, in some, it is recognized that that information may not always be available. So, well, you need to, to get some more general approach. So even the guideline makes its own life difficult in relation to this this request. The authors of the guideline and some other colleagues have looked then later on, and 4 years, it's not so much time after, to the outcomes of the use of the guideline in 2001 and 2005. They have published the first, let's say, paper where they have analyzed the impact on safety pharmacology guidelines in drug development. This is the paper. And Olentizar, for instance, he has been one of the authors of the S7 guidelines, and especially very much involved also in S7B. which is for the cardiovascular system. So the authors there, they have recognized that the guideline was doing well, but anyway, there were areas that would need further requirements, and which would be, of course, selection of doses and concentration and the choices of species and the modeling of the temporal pharmacodynamic changes in relation to the pharmacokinetic profile of parent drug and metabolite. I would say that this aspect here is quite interesting and important because somehow it looks like looking at the studies which are conducted using one dose or few administrations, and they are recognizing that this might not be sufficient to cover the pharmacokinetics and then the metabolism and the timing for all this to happen, and therefore they even are recognizing here the limitations of the let's say the studies by themselves to be conducted the way they are being conducted according to the guidelines. Then they say, while it is recognized that they could mimic through these few administrations, few doses possibly, what could happen in terms of safety concerns for phase 1 and 2 volunteers and patients, it's not clear that this guideline would, and the studies conducted according to them, would be quite useful to cover the safety during the later stages of development or when the drugs have been incorporated in the market. And I would say this is quite relevant. And this is quite relevant because we see more and more that drugs are developed according to their action over very specific targets, very often very human specific, where the cascades are well identified, where distribution is well identified, the pharmacology is well identified, and somehow, well, you expect and you see many effects that even are responsible for withdrawal of drugs later from development or from the market being related to the pharmacology. And if the pharmacology, so that means somehow safety pharmacology, and if safety pharmacology studies have been conducted, but they have been conducted just to cover very early effects that might come from few administrations, of course, that I would say that we are driven to think that what we see with products and pharmacodynamically driven effects is not sufficiently covered by safety pharmacology studies. So something might be needed to take better profit of these studies, which indeed will cover pharmacologically driven safety aspects. And this is safety pharmacology somehow. So putting into context from S7A into the more recent guidelines, where the ICHM3 guidelines have been revised to adjust to some, I would say, modernizations of development, where S6 for the biologics has been modernized also and updated in the same context. And as S9, the guideline that refers to requests for preclinical studies for the development of anti-cancer drugs, we really see that let's say the guidance itself drives into the direction of supporting that incorporation of safety pharmacology endpoints in toxicology studies might be of value. You would see that for the biotechnology derived products, while thinking that this incorporation might come because, well, these studies are conducted mainly in non-human primates, so you need to save non-human primates, and therefore you can put together toxicology and pharmacology. On the other hand, you see that with this, you are able to get the studies conducted in the relevant species, and at the same time, you are able to capture endpoints that could be seen along the time, and therefore after more persistent pharmacology. So somehow they might be more, let's say, in line to what is needed than the purely conducted safety pharmacology studies, very early conducted just to support the safety before first in human studies. And I would say that somehow this goes into the same direction for the anti-cancer drugs. And if we think on the timing, again, an evolution has been seen. While ICHM3 was saying, okay, you just need safety pharmacology before first in human studies, so whatever is the study, you need safety pharmacology before going there. Now, ICHM3 R2, the revision, has incorporated new types of first in human studies, which might be exploratory studies, which could be conducted with very low doses, for which even the pharmacology might not be relevant because of the extension, the dosing is the dimension of the dose is too low, and therefore they are not recognized as being needed. So there is a type of first-in-human studies not needing safety pharmacology before, and when safety pharmacology stops being requested before first-in-human studies is when you are approaching doses in these studies that will cover or will be subtherapeutic doses, using subtherapeutic doses, so close to pharmacology effect. And there, at the same time, these studies need to be supported or by toxicology, extended toxicology, or by 1-week toxicology studies, where safety pharmacology is requested to be conducted with the dose that would cover the toxicity studies. So somehow you need safety pharmacology and you need toxicity, and the doses should be the same. And again, the question is, or at least should be covered. The question is, okay, then why doing 2 types of studies? Why not incorporating teaming safety pharmacologists with toxicologists, putting them together, looking at the studies, and maybe looking at the same studies and capturing the data they need to take their conclusions? Maybe it could be better use of resources, and at the same time, even the teaming and the crosstalk between those type of specialities could take more benefit of what the data that is produced. So somehow I would say that looking at this evolution of the guidelines, we can only conclude that the idea is being reinforced that on the possibility to incorporate the safety pharmacology in general toxicity studies, together with some advantages that are recognized. And just to give the support for this thinking on this aspect of the safety of compounds being very much related to the pharmacology in many situations. For instance, let's think about this torcetrapib, and I have that example because I have built this some time ago. There are many others where this is a cholesterol transferase protein inhibitor, anticholesterolemic drug, and this drug has been withdrawn from development at a late stage, phase 3, just because cardiovascular events, because related possibly with hypertension, and with some, which is related somehow with their pharmacology. And the question is, what are you going to do now? Would you have captured that if you had incorporated safety pharmacology in the toxicology study? Or are you going to do that now? That's not the same class, just trying to capture that. And I would say this goes into the direction that really these situations could justify. There are situations that clearly point towards the need. The question then is whether you should make it as a principle, or you should make it in some situations when the need is identified. And that's what is written here. Do we think that since ICHM3 says that, yes, when possible, you should do it, and it could be justified, incorporating studies in the toxicology, what's the way forward? Should we think that this could be the default position? You should think about that. And then in selected, justified situations, you could make separate safety pharmacology studies, or should it be the other way around that it's going now? I know that this is a challenge for the institutions that are separately developing safety pharmacology studies and toxicology. Maybe this is the adjustment that might be needed. This might be the rethinking of some of the institutions and the way people are working. But, well, that's what we are doing all the time, isn't it? I came from my university this morning, and the big drama is exactly that one, trying to change a bit the structure, incorporating in the different way the talk between the different scientists who are there. So I think that's life currently, that we need all the time to rethink, and to rethink not only based on ethics, because I think the worst argument, in my view, that we can use now is to say that we need to do this or that because we need to spare, to save, and to protect animals. I think it's the wrong argument, because it can be bought by some, it wouldn't be bought by others. But when we think that science really justifies and is pointing towards that direction, I would say that at a certain moment, the ethics is really the fact, justified by the fact that the approach is more scientifically valid. So, and I think that's what needs to be put into the perspective and in our mind. On the other hand, there are multiple innovations. What the reality when ICH-M3 has been put in place, or when safety pharmacology in the 90s or the 80s even was conceived, is not the same reality as we have now. Currently, most of the drugs are being developed towards a certain target, which is well defined, is well known, which is well known in terms of its functions in different tissues, in different systems, in human systems, in animal systems. And therefore, based on target distribution, target systems, cellular cascades associated, the crosstalk between cascades, it's much more possible to anticipate the, let's say, a spectrum of pharmacodynamically driven effects that could be occurring when using the drug. So somehow there is much more based on in silico systems than to be confirmed in terms of in vitro methods that currently more and more start using even not animal but human cells, therefore getting close and close from the target species to which the drugs are addressed. So on the top of all that, technology available now is allowing that telemetry so invasive or non-invasive systems could be incorporated in the toxicology studies just to be able to capture the endpoint. And I would say now that going even beyond that, this is something that is even being explored as new strategies to monitor patients or patients in trials or patients in real life. This aging society where more and more you will need to monitor aging people at home, and then you need strategies to capture their glycemia, their blood pressure, their behavior or whatever when they are in their own environment. So somehow, this, let's say, the concepts, they go even beyond the animals. They start being used for humans. So somehow they become more and more logical and more possible. Yeah, this is exactly the point. The medicines increasingly target specific, as I said, and this is like then the need to change. Is it a wish? Is it a need? The fact that the pharmacodynamically driven effects cannot be always predicted after one or few administrations. So I would say, drive us into the direction of thinking that, yeah, most adverse events are related to pharmacology. You cannot predict it after a few administrations, emerging in repeat toxicity studies, and sometimes the effects emerge there, and then you have to make mechanistic studies just to look at that function. So somehow this is becoming more and more the rule, I would say. And on the top of that, I would just, before finalizing, also highlight the fact that these predictive tools that are becoming more and more available, that displace us even from animals and focus more on human systems, using human cell approaches, they are really, human cell systems, they are really becoming a reality also, which will allow us to understand the pharmacokinetics even before we have the first human study. This is a reality also. And we have the systems pharmacology, and we have the systems physiology, which we have the modeling that allow the PBPK modeling and relationships to be established, and all these put together in systems that are obtained from human cells. Because really the science and the technology around the pluripotent stem cells is allowing that micro mini human organs are starting being collected, used, and therefore there is information that we start collecting in vitro, in silico, and then confirming more or less in human systems. And this will become reality, and possibly in the next decade. So how are these advances then impacting on the discussion we are having here of having the possibility to really incorporate the endpoints in animals or in living systems? Just to give the example of the lung on the chip, of course, you I'm sure you know about this, but yes, this is the best or one of the best models that we can now start looking at when you see this lung where you have the blood flow and you have it breathing and you have even the inflammation, capillary, whatever, everything is in place somehow, and this model is breathing. So, well, and it's the start. It's becoming now being available, and more will be adjusted, and by the end, when you have available a lung in a chip, and a heart in a chip, and a small intestine in a chip, and a kidney in a chip, which, by the way, are being developed at a high speed, I would say, particularly in the U.S., through the NCAS and through the Vice Institute. So I would say that at a certain moment, it's possible that the anticipation of in vitro and in silico could be confirmed in systems which are human systems. And now, then next step is where are you going to confirm this safety pharmacology aspect? Will it be in animals? Will it be in humans directly? I don't know, but specifically it doesn't make such a big sense that you will need separate in vivo studies to address this when you will have all these models available. I would say that to finalize and reflecting, the guidelines that we have in place, they are clearly, when possible, favoring incorporation of these studies in toxicology studies. For the biologics, yes, that's the lessons we got because it's more targeted related, more pharmacodynamically related, but it's becoming a reality for all types of molecules because more and more they are really very pharmacology and target focused. And then I would say that the experience, either we want or not, seems to be applicable. I would think of glycosurics, for instance, these anti-diabetic molecules blocking the transport of glucose. Yes, I would expect pharmacology on the cardiovascular system and renal system to be affected. Where do I look at this? Do I go to toxicology studies? Do I look at it as safety pharmacology studies, or do I put both together? And this is just an example. So the challenge, I would say, is whether we would think that this is a process that goes progressively and naturally will occur, or whether there will be a need of reinforcement of some guidance that will better allow to go in this direction. My view is that there might be no need for new guidance to be in place. But yes, I think this is one of the aspects that might deserve some further discussion. And that's all. Thank you.
Great. Well, thank you so much, Dr. Silva Lima. That was a really fabulous overview of where everything is going. I'm going to actually ask people to, or remind people to put your questions into the chat box. And in another moment, we will also put up a questionnaire, a survey, for you to fill out and give us feedback on future webinars and discussions that we'd like to hold. So we want to really encourage everyone to send in questions, but I'll start with the first question here. And it is for you, if one wants to incorporate safety pharmacology in a tox study, let's say a 14-day tox study, when should the safety pharmacology assessments be performed? After 1 day or after 14 days?
Well, I would say that based on, let's say, the reflections that I was developing along the study, I would say along the presentation, and the more I think about that, the more I think that it might make sense that, and it could be wise to have, let's say, an initial evaluation and then a late evaluation. If you ask me whether I think it would be better to have it 1 day or 14 days, I would choose the 14 days. But anyway, if you want to support a first-in-human study, it might make sense to understand which could be the consequence after a first or a shot, I would say, a first administration. But safety pharmacology should go beyond that. So I would favor very much the measuring also after long term, so after at least the 14 days. 14 days, or at least it's the 14 days, or it's the let's say, the moment where you have the kinetics stabilized. So yeah, at least the 14 days.
Okay, thank you very much. So again, I'll encourage people to put questions into the chat box. But here is another question for you, Dr. Silva Lima. How do you see in vitro systems replacing safety pharmacology studies in vivo?
In vitro systems are being developed now. We have already some in vitro systems. Safety pharmacology is one of the examples where in vitro systems have been accepted or put in place to address some aspects of a system, cardiovascular. Specifically, if we look at S7B, we see that the several in vitro systems together with in vivo, electrocardiogram, whatever, but several in vitro systems are considered in capillary muscle, in ventricular muscle, in her channel to address potential proarrhythmic effects or potential electrophysiological effects in the heart. So it's a good example of in vitro possibilities. So now, if we think that, in addition to that, we have the possibility, for instance, to address gastrointestinal, going into contractility, for instance, of gastrointestinal muscle, or so some of these aspects have been already taking the profit of in vitro or ex vivo systems. If you think now that you might have, when you have developing micro-human organs that could be mimicking and working like a kidney or like a heart with the calcium, with the channels, with the receptors in place, or even having to put multiple systems where you will have all this integrated. If you think that you may have a life where you could have the breathing, you could have the inflammation and the monocytes, and yeah, even the immune reaction happening, the recruitment of monocytes after inflammation or whatever, these things are happening. So I would see that these systems, these human systems based on human cells, even human cells that could be collected from healthy volunteers or from patients, could be enormously useful to mimic human situations and to anticipate which could be the consequences of the pharmacology of the drug on these multiple systems, which, by the way, now are being already planned to be connected because this Vice Institute, together with NCAT, they are already announcing that within some couple of years, they will have at least 10 systems connected by microfluidics, which means that you could have simplified human in chips. That then the discussion is, what is more reliable for humans? Is it a mouse or a rat or a dog or a monkey that you partially look at, or is it this, let's say, simplified human system? And well, it will evolve, of course, but the point is it's not ready to be used possibly, but this might be the future. So I see really the in vitro in this perspective a very powerful tool for the future.
Great, thank you. We have another question here. Great, thanks. So what do you think the necessity of safety pharmacology studies is for ocular drugs before IND filing?
Ocular drugs. Well, safety pharmacology is related to the pharmacology on vital functions, isn't it? So if you have an ocular drug and you are concerned, you need safety pharmacology, I would say that you need safety pharmacology. You might need, if you have a concern related to any systemic absorption of the drug at relevant concentration that will could lead to some pharmacologically driven effects to happen systemically. And this means that you really may be able, in my view, to justify that it might not make sense to conduct a safety pharmacology study for that drug if you show the pharmacology, if you show that even if there is systemic excursion, that the systemic excursion might be very low, you should take into consideration that, for instance, you can connect to ICH-M3 or to where for microdosing studies you don't need to come with safety pharmacology. So by establishing these bridges, you could justify that you don't need safety pharmacology. So I wouldn't embark in conducting safety pharmacology before I could justify whether it would make sense or not. If it doesn't make sense, you shouldn't conduct it.
Okay, thank you. So another question here is, in this safety pharmacology studies of a small molecule oncology drug, in your opinion, is it essential to use jacketed animals and or telemetry for monitoring their response to the drug?
For a small molecule for oncology? Well, I would say that I wouldn't detach that decision on any other type of drug. The S9 is clear. It says that you can incorporate safety pharmacology in oncology. You can incorporate for oncology drugs. You can incorporate in vivo in toxicology studies, and you might not need even for the first human studies. I think the S9 says that. And in case but then you need safety pharmacology. If you need safety pharmacology, why not making the telemetry, yes, and conducting that way? So with the incorporation, why not? Okay. So I would say I would keep this difference from at the moment you decide to make the safety pharmacology. I wouldn't see it different from another situation. And in the incorporation for the toxicology, in the incorporation within a toxicology study, and then in the methodology that you might use. Okay, great, thank you. We have another question here. In the case of incorporating toxicity studies, what will be the impact of this in the toxicology studies and the impact of toxicology studies in safety pharmacology? Yes, of course. I think that's the question. I think that's exactly the question. What do you think that, what is the consequence for the toxicology and what is the consequence for the safety pharmacology? And that response somehow I would say it's to be responded in line to the outcomes of the experience with the biologics. Within the biologics, I don't think that the fact that the animals could be monitored for their blood pressure or for the respiratory or whatever would have this great impact, because what we think and the big concern is normally the stress related that could or exacerbate the pharmacology or the other way around, or could exacerbate the toxicology. But if we think that the toxicology is mainly related to well, after a certain moment you have, biochemistry and histopathology, and pharmacology is functional endpoints, I would see maybe the safety pharmacology at the moment and specific toxicity happens, I would see that the functional aspect might be impacted, particularly the stress, the CNS, and then cardiovascular, respiratory, or whatever. But I have some difficulty on thinking that getting safety pharmacology endpoints in a toxicology study would impact so much toxicology. Somehow, it's also, in my view, the way we are looking at toxicology studies. We really want these toxicology models, which are these healthy animals, which are treated with the drug, all animals the same. So being very much standardized and reproducible and whatever, as much as possible for the toxicology. That's why we are so afraid of changing let's say the environment, or adding some measures or so on. But when you have a control and you can follow the control also, I think that it will be possible always to have some notion of the impact on these measurements on both sides, pharmacology and toxicology. And it could be that it might have some impact, but you already have this done with the biologics. And humans, in general, that's what we want to predict. Normally, they have to be subject to both, isn't it? To the drugs, to the treatment, and to the measurement of the effect. So somehow it might be more realistic.
Okay, great. Thank you very much. Another question for you. This is a more detailed question. So in your opinion, if one develops a pediatric drug only, should the safety pharmacology studies be done in juvenile animals?
Oh, that was a very, that's the question that we have been debating from ever. I remember when at the UK we were with the Royal Academy of Medical Sciences discussing in an integrated way the value of development of animal studies. This has been exactly one point. How do you do with pediatric drugs? And well, I would say that going to safety, well, pediatric drugs and juvenile animals, that story is not yet, I would say, complete. I don't think any of us can say that we are really happy with the outcome of juvenile animal studies to predict the outcome, I would say, juvenile or pediatric toxicity. There is a lot of effort to be able to find models, juvenile models that will mimic the pediatric situation. And when we think in global, so when you see and when you think that you have the different systems developing at different paces and at different time lags, so it's very difficult to put a safety pharmacology study to address several endpoints in a juvenile animal, unless you are going to a juvenile animal to test for, let's say, because safety pharmacology is about different systems. So if you have a respiratory system, a juvenile dog will develop it at different pace as a baby, as a rat. The kidney is the same. The cardiovascular, I don't know. The immune system is also different. And at a certain time point, if you really need different systems to be addressed, you will need different models, juvenile models. I would start thinking that this might be too less feasible. And then we have the discussion on CNS and the behavioral aspects and so on. And again, the real value of the juvenile to address all these aspects is complicated to conclude. So I wouldn't, we still don't know the value of juvenile animal studies to address even the developmental aspects in general and the impact of a drug on developmental aspects in general, I would be even more concerned if I would have to mimic the pediatric situation in juvenile animals. So I don't think that it's sufficiently feasible, I would say, to use juvenile animals for safety pharmacology studies.
Okay, thank you. Another question here is, in obtaining safety pharmacology study data on respiration for an oncology drug for use in an IND submission, would it be okay to monitor the two endpoints, respiration rate and O2 saturation? So SpO2 pulse oximetry.
Well, I would say yes, but I would go beyond that, and I would try to understand and that they. So the mode of action. So if you want to monitor respiratory potential respiratory effects, I would say that it might make sense to look at your drug specifically to the mode of action, potential impact, specific impact on the respiratory, and to adjust even the endpoints together with the general endpoints, which are put in even in general the guidances and so on, to see whether your drug would need some more attention on one or another endpoint. If you are going to a drug where you don't have respiratory concerns in general, so I would say that the normal endpoints for the respiratory studies would be sufficient. And then when you go to the clinical trial, maybe you could do a parallel evaluation also if you have a concern.
Okay, thank you. So another question here, and again, I'll just remind everyone that the survey is up. If you could take a moment and fill out the survey and send in your last couple of questions, that would be great. So what will be the place of safety pharmacology in the drug safety assessment in a future toxicology paradigm?
Yeah, that's a very good question, because somehow the question is, what is the place? Somehow we have to start by thinking, what is the place of safety pharmacology in the current paradigm? Safety pharmacology in the current paradigm is a kind of a reassurance for the administration of humans, early administration, particularly to healthy volunteers, and then in some situations to patients, for the early testing, isn't it? So that's where you need the safety pharmacology, and that's why when you go to regulators, sometimes later on, and you have already had some human experimentation, some clinical trials, where some safety pharmacology studies or endpoints were missing, you can justify that you don't need to come back to animals to conduct those studies, because these might have been already covered by the human data that you have generated. So, having said that, I would say the safety pharmacology, the role is more to predict early administration, the way it is being used now. But what we see now is that a lot of adverse events that emerge later in drug development or at the marketing authorization stage, in the post-marketing, and have justified even some withdrawals, could be related also to functional effects of the drug, direct or indirect, so related to the pharmacology. Directly or indirectly, we see some effects on pharmacology. So I would say that if safety pharmacology has a place in a future paradigm, I do believe it has. I would say that it has an expanded place, an expanded role that would allow, that would be addressed the understanding or the prediction of potential adverse events related to the pharmacodynamic effect of the drug. So thinking about that, it means that you might need safety pharmacology studies that will allow to mimic potential functional effects after an administration that would go similarly to human administration, so long-term, whether you need that in animals or whether you could anticipate in silico, in vitro, taking into consideration what you know with systems biology, pharmacology, and so on. Then mimic the systems in human systems the same way we would make some of the studies in vitro. And then to confirm in vivo model integrated, that could be even the human model. And well, we have the example of the solidity, which is somehow a safety pharmacology study. It is. So it's an example on how it could be anticipated. So I would see the safety pharmacology with an expanded place, because it will really address pharmacodynamically driven safety. And whether it will continue to be conducted in non-human models or it will move towards human models, my feeling is that it will move towards the human model.
Great, thank you. I think we have perhaps one more question here. So if non-rodent is a more sensitive species than rodent in terms of toxicity, should we conduct safety pharmacology studies in non-rodents?
Well, I have difficulties with well, I think it's not a yes or no response, because non-rodents, oh, no, sorry. You are saying if non-rodents are more sensitive, in case for a drug where non-rodents are more sensitive, should you conduct safety pharmacology in non-rodents? I would say that not necessarily. If you can have, let's say, if you have to choose between if you ask me that as a regulator, now I'm not a regulator. If you ask me that as a regulator, I would say, okay, you have a very sensitive model, which might be the non-human primate. You have a rodent that is less sensitive. It's a matter of adjusting. And doses could be a matter of adjusting conditions in order to predict whether, in case an effect occurs, which effect would occur. I must tell you that I remember some discussions previously while I was in my laboratory environment where we had exactly that point. There is a non-human primate which is more relevant and more sensitive. There is a rodent which can be used. Being less sensitive, what do you use, a rodent or a non-rodent? And we ended up by saying, well, you can use the rodent provided that you use a system and you use a protocol that will show sufficient sensitivity. You might not need always the most sensitive species when you have to jump into, let's say, ethical problems, high species use or whatever. So you could justify a lower sensitivity, provided that still what you will get, you can show that it's relevant.
Great. Well, thank you very much, Dr. Beatriz Silva Lima, for your presentation today and for answering all these terrific questions. We really appreciate everyone joining us for this webinar. And as you all know, we are recording it, so we'll be sending it out to you afterwards to listen to and share with your colleagues or for anyone who missed today's presentation. So again, thank you very much, Dr. Beatriz Silva Lima, and thank you everyone for your participation today.
Thank you for the invitation, and thank you to those who have waited until the end. My pleasure.
Great. Have a great day, everyone. Take care.
Thank you. The same. Bye.